Let's talk today about cell division, all right, how cells divide. We're going to focus on something called mitosis, but this is one way the cells divide. Um, not all cells will do exactly what we're going to go over today. Then we're going to compare it to meiosis. Okay, meiosis is similar to mitosis, you'll see, a little bit different. This is specifically about eukaryotic cells. So bacteria they're going to reproduce a little bit differently because if you remember, bacteria don't have a nucleus, and that's actually going to affect the way cells divide. All right. So we're going to talk about how cells divide in eukaryotic cells. All right. Mitosis is what we're going to focus on. Okay, so here we go. The idea in cell division, big idea, is we have to ensure that we copy all of the DNA. So if you remember, the DNA is the programming, right? The set of instructions in the nucleus of a cell that allows a cell to pretty much, carry out, execute its functions, right? This, DNA will control things like we talked about last time, what type of cell it becomes, depending on what genes it uses. The DNA will instruct on how to build certain proteins. It will give you your characteristics. It will, control things like cell division itself. So like the programming itself controls stuff like when does a cell divide? When does it not divide? These are things that are all regulated by the DNA in the nucleus. So this is very valuable information. It's organized into these things called chromosomes, which you can see here on my image. These are just stained chromosomes, so you can see them. They will look like this. This is probably a structure on the left that you might be familiar with, that famous X-like structure. These are the chromosomes when they thicken up and they condense. So they actually don't look like this normally. They'll only look like that when a cell is going to divide. If you zoom in, the chromosomes, which look like an X, are just really loops of string. So you can see on the right, just like looks like a string that's been looped. That's your DNA, okay? That DNA is wrapped around, so here are the loops, these on the right, proteins. Okay, you can see here's another picture of that. So the DNA that looks like a loop on the right is actually looped around these proteins called histones. You can see on the right, in here is a black and white image. You can see the actual histones, so like little circles. So when DNA is copied, it needs to be also sorted out. So what's going to happen to the DNA is in cell division is it's going to be copied. So we have two copies of it. This is what it looks like. And then it's going to be sorted out. And I'll explain what that means in a moment or so, okay? Here's another image of that. So if you keep zooming in, you can see there's your DNA structure. They're wrapped around those proteins, which again, I told you they look like this when on the left, when chromosomes are about to divide. And we're gonna talk about that during cell division. Okay, so let's take a look at how this works. So if you remember last time I showed you a video, which was the inner life of a cell. So actually, let me go back to that video. It was a very nice video. So I want to show you a specific part of that video. I think it's here. Okay, there is a specific part I want you to notice, which is right about that right here. Okay, so this is, if you remember, this is the cytoskeleton. And you can see here it's forming. Here it's forming, it's getting longer. But then you can see here it's breaking up and getting shorter. Okay, now the reason why I'm showing you this is because the cytoskeleton is going to play a very important role in cell division. You're actually seeing it right here, okay? What you're seeing in this image is in the middle are chromosomes. We know chromosomes are basically, that's your DNA, it's right there. And you can see that all that green fiber is attached to the chromosomes. And you saw in the video that these fibers can get longer and they can get shorter. What these fibers are going to do, they're called spindle fibers. Here's the term right here, spindle fibers. They're actually going to help organize the chromosomes so that we can separate them, okay? You can see that in this picture here, right? This is just basically the same image as this. Just obviously, this is just a little bit easier to look at. So you'll notice that here is a cell, and you can see chromosomes are like lined up in the middle. Now the chromosomes, I said, are basically your DNA strands, right? They're the DNA strands that again are wrapped around proteins, and you can see that they're all lined up. Okay, now why do we have to line them up? That's this is an interesting question. Like why, they're all like line up right in the middle of the cell. That's obviously something important going on there. We zoom in on a chromosome, there you could see there's that famous X-like shape. Remember, this is just really DNA, right? That's what that is. It's DNA that's looped. So in there are sets of instructions, on how to build insulin, how to build hemoglobin, how to tell a cell what cell to become, when to divide, stuff like that. Okay, so what goes on during cell division? That's what we're going to look at. All right, so let's actually just jump down to cell division itself. Let me skip over this. We don't need to look at this right now. Okay, so there are certain steps in cell division, right? When a cell divides, it's going to happen during certain conditions. So when does a cell divide? Cells are going to divide when an organism is growing. So for example, if I have an organism that is really tiny, maybe like the beginnings of like a human embryo, the first thing is, if you remember, just one cell, right? The zygote. Let me clear the canvas. So when human life begins, we have a cell, which is called a zygote. Now, what happens is that cell is going to make a copy of itself. So instead of having one, you'll have then two, and then each cell is going to make a copy of itself. So then you'll have 4, right? And then 8, and then so on and so forth. I'm not going to keep drawing, so I'm not really that good at drawing. But each of these cells has the DNA in it, right? Which I'm going to try to draw as red. Now, what it's going to do is going to make a copy of that so that each of these cells will have the exact same DNA inside it. So during growth, that's when cell division happens. Also, if there's an injury, like if you get a cut in your hand, so I'm going to try to draw a hand. Imagine this is a hand. Now, if you get a cut, imagine you get a cut, right, in your hand. Now, we know that over time, the cut will just disappear, right? Maybe you're left with a scar. Now, what happened in that case? Well, what happened in that case, if you were to zoom in on the cut, imagine the cut is like a gap, like this. Well, the cells in that area, which maybe I should draw as black, these last time, they'll divide. And as they divide, and they'll fill in that gap. So, cell division is really important for growth, but also for repair. Two very, very important functions in cell division. Okay, so, but if it's going to divide, it's going to have to prepare itself for division. So there are two kind of phases that we want to look at. One is like a preparatory phase. It's going to prepare itself for division. And that's interphase. So interphase is, you can think of it two ways. One is you can think of it as the prep, when it's going to prepare itself for cell division. You can also consider it the rest stage, because either a cell is going to be dividing or it's not going to divide. So it might be just a rest. Maybe the cell is not dividing at all, and then it's set to be an interphase. But if the cell is going to divide, it's going to need to prepare itself to do that. So interphase is also when it does that. It's either going to be resting or preparing itself for cell division. And then when you have cell division, that's actually the M phase, which the M stands for mitosis, which is the actual cell division itself. So what's going on during this prep phase? Well, if a cell is going to make an exact copy of itself in interphase, which you can see, think of this as a clock. You can see the life cycle of a cell is mostly during this interphase. This is, again, the rest phase or the preparation phase of cell division, right? What I want to focus on is this part right here. This is synthesis. What's happening during this part is if a cell is going to go through cell division, it's going to make a copy of its DNA. So it's going to copy its DNA. So what that means is if you have, for example, in a typical human cell, there are 23 pairs of chromosomes. So if I go back to what a chromosome look like, there will be 23 pairs of this, okay? Now, when a cell divides, it's going to copy everything. So you'll have 46 pairs because you're going to double everything. The reason why we do that is because each cell is then going to get 23 pairs, right? So mathematically, that kind of makes sense. If I take, for example, let's clear the canvas. Imagine this is my cell, and it has 23 pairs of chromosomes. That means it has 46 chromosomes total, right? By the way, I should mention why 23 pairs? Well, because if you remember, the zygote is formed from two cells, a sperm cell and an egg cell. And when they fuse, they make a zygote. And what happens is each contributes 23 chromosomes. So the sperm would contribute 23 chromosomes, and the egg would contribute 23 chromosomes. So mathematically, that adds up to 46. We call them pairs because you get one set from dad and you get one set from mom. So they're paired up that way, okay? So when that cell divides in your body, what's going to happen is that cell is going to make a copy of everything. So instead of having 23 pairs, it'll have, I guess you could say 46 pairs or 92 chromosomes. So when it makes a copy, it'll end up having 92 chromosomes, okay? Initially had 46, because then when it divides into two cells, it's going to split those chromosomes back so that it has the same number as the original cell does, right? So that these two are identical. So this is really important. This is going on during that preparation phase, that interphase, preparing itself for cell division. Okay, now that's a really important. That's the only thing that happens. The cell also grows in size. There's a bunch of checks that it does too to see if there's even space for growth. But let's just focus on the DNA. Okay, so let me jump down a little bit. We talked about the purpose of all this. So this is what it looks like. Imagine this is your cell. So in the nucleus of the cell, you have your 23 pairs of chromosomes we talked about. In other words, 46 total. 23 pairs, 46 total. Now, what we said was that, the chromosomes, the DNA, everything's going to get copied. So you'll end up having double of that, 92. Now, during this process, when the cell is preparing itself, the chromosomes actually become visible. Normally, you can't see them. Like if I look at this picture here, I don't see any chromosomes. I just see like a bunch of purples, spots and stuff like that. During cell division, the chromosomes actually will become thicker and you can actually visually see them. Now, you'll notice in this image, not only can I see the chromosomes, but it looks like the nuclear membrane is starting to break up. You notice there's like gaps now? Okay. That's actually very important because if I'm going to arrange the chromosomes and move them around, I have to get rid of the nucleus. Otherwise, how am I going to do that? The nucleus is going to be in the way. So I'm going to have to get rid of the nucleus. So that's why you're seeing those gaps. In this image, you're also seeing the copies of the chromosomes. You'll notice that these two pairs, or these two sets, call them sets, are the same length. You see how they're the same length? And that's because they're, so notice one's red, one's blue. Okay, so let me quickly explain that, what that means. So it goes back to what we said before. One set comes from your mother, one set comes from your father. So let's say the red is the paternal one. That means from the father. And let's say the blue is the maternal one, okay? The set from your mother. Which would mean, if I look at the smaller chromosomes in this slide, so that would, if I said red is paternal, then that would be a set from dad, and then that would be a set from mom, okay? Now, you'll notice that the X-shaped pattern, the reason why it's an X-shaped pattern is because the DNA got copied. So instead of it looking like just like a line like this, if I copy that chromosome, it actually now looks like this. This is my copy. So it gives you that X pattern. So in other words, what this means is that these two strands, so this strand here and this strand here, are copies. Okay. We can say the same thing, for instance, these two. So these two would be copies. I said these two are copies, these two are copies, and these two are copies, which is what I said before. If a cell is going to divide, it has to copy all of its DNA. Now, I have an animation to show this. So for instance, here it is, okay? So here is the, copying of the DNA. So if you notice right now, you see just two strands, okay? So one's red, one's blue. That represents one from your mom, one from your dad. And then what happens is the DNA gets copied. You can see that there, right? So now we have two strands stuck together. And because you made a copy of everything, so now you made two copies of that one strand for each chromosome. So I said, mathematically, if you had 46, now you have 92, for instance. Okay. So this is what's happening during interphase, right? It's got to prepare itself for cell division. Now, you'll notice what happens is the nucleus is starting to break down. And you can see here that it's almost completely broken down. And what you're in right now is actually mitosis. So this is actually the part where the cell is actively going to go through cell division. Okay, so what you're also noticing, and what you didn't see in this slide, is these black fibers. Notice this? So these are your spindle fibers, part of your cytoskeleton. Notice that they're growing. They're looks like they're getting longer. It's kind of hard to see it here. I don't know if I have it in this animation. I might have it here in this animation. So you'll notice in this animation, the spindle fibers are growing. So this is prophase. Now, if I back up a little bit, see how they grow. And the difference between prophase and metaphase is the location of these chromosomes. So in prophase, they're not lined up in the middle, right? You see that? And then we have metaphase. And you can see, so meta means middle. So during metaphase, the chromosomes are actually lined up in the middle. And you can see that in this slide here. So they're all lined up in the middle. Now, why are we doing this? Why are we lining up in the middle? Well, because every cell is going to, every daughter cell, we're going to call them daughter cells, right? So this one cell is going to split into two daughter cells. Those daughter cells are going to want an exact copy of the information. So how do you get an exact copy? Well, you have to organize the DNA. So you're going to line them up in the middle so that we can evenly split them. So that's what you're seeing here. So in metaphase, the chromosomes are all lined up in the middle so that we can evenly split them. See how they're getting split? Now, when the chromosomes get split, then you're in another phase called anaphase. So there's actually 4 phases to mitosis. The first is prophase. That's when the chromosomes are, the nucleus is gone and the chromosomes are not quite at the middle. The spindle fibers are starting to reach out and grab them. And then prophase is when the spindle fibers has moved the chromosomes into the middle. And then anaphase is when the chromosomes shorten and split them. So they're literally pulling them apart. And you can kind of predict what's going to happen next. So the next stage is something called telophase. And that's when the chromosomes get pulled to opposite ends. And again, you can see now why we need to line them up so that we can evenly split them. And you could see that right here. So there's your metaphase, and then there's your anaphase. You see the chromosomes getting split. What's going to happen? You'll notice the cell also looks like it's it's quite circular here, but here it looks like it's getting stretched out. So at the end of telophase is a process called cytokinesis. And this is when the cell will pinch in the middle and split. And you can see what happened. By doing that, now we formed 2 cells that are identical to each other. And they're identical to the original cell. You can actually see that in this image here. So there's an image of cytokinesis, that pinching in of the middle, so that you end up getting 2 daughter cells that are exactly the same. Okay, so just to review, so interphase is the process of preparation, and then you have mitosis is when the cell is going to divide. And mitosis can be divided into 4 stages. One is prophase. So prophase, that's when you can see the nucleus is breaking down and the spindle fibers are elongating. Then you have your metaphase. That's when the chromosomes are in the middle. I think M for middle. Then you have your anaphase. That's when the chromosomes are getting split to either sides of the cell. And then you have your 4th phase is your telophase. And you can see what's happening in telophase. The nucleus is coming back. You notice that? You see it here? The nuclear membrane is coming back. And in this slide, you're also seeing the cytokinesis, and that's the cell splitting in the middle, because it's going to pinch in the middle and form two cells. So, I have some slides here. For example, here you could see this in plant cells. So you can notice that, so here's a cell right there. There's a cell. Now, plant cells, if you remember, have a cell wall. So they can't pinch in the middle because the cell wall is very, very hard. So instead of pinching in the middle, what they do is they put up a wall. You can see that wall right here, okay? And that wall is called a cell plate. Let me see if I can make it a little easier to see. Okay, it's called a cell plate. But essentially, it's the same idea. The cell's got to split at the end of cell division. Okay, and again, that process is called cytokinesis. But in plant cells, you can't do this. You can't pinch in the middle. So what you end up doing is you form a cell plate to divide the cells. Okay, that's a bit of a difference there between the two. But that's mitosis. And by the end of mitosis, you end up with two cells that are exactly the same. Okay, exactly the same. And we talked about the difference in plant cells. Instead, you get a cell plate. So you can see that here. Okay, cell plate forming. But in the end, you get same thing. You get two identical cells, right? Here's kind of a nice, beautiful image. You go from the left all the way to the right. You can see the different stages of cell division. You could see the cell as we go through this. You could see the cell, the chromosomes are on either end here. You can see the cells splitting in half, and then you end up with two cells that are identical. Okay, very nice, beautiful image here. Here's a wonderful image of, you can see anaphase. So you can see the chromosomes being pulled to either end in this beautiful image here. All right, very, very nice. Chromosomes in this case are in blue, so you can see they're being pulled back to either side of the cell. So, if we want to test ourselves, for example, like this would be, what do you think this would be? What stage would this be? So these are all stages of mitosis. So it's not interphase, okay? So it's either going to be prophase, metaphase, anaphase, or telophase. So P, M, A, T. What does this stage look like? What do you think? Anyone want to guess? No one wants to guess? So this is prophase, because you can see that the chromosomes, you can see the chromosomes, but they're not lined up in the middle, right? And they haven't looked like they've completely separated. Here, this would be anaphase, because it looks like they're being pulled to either end. So that would be anaphase. This would be interphase. So, so I lied to you. So this is actually not a stage of mitosis. This is interphase, because you can see the nucleus, but also you can't really make out the chromosomes. So this would be, but this, you could argue that this is the beginning of possibly prophase, right? You got to remember one thing about these stages. Sometimes the line between one stage and the other stage is very, very blurry. So what I mean by that is, like, for example, like I know today is Wednesday, and I know tomorrow is Thursday. But there comes a point where, like at midnight, we'll get to that boundary between Wednesday and Thursday, where it might be a little bit fuzzy. You can think of like there's a specific, maybe a brief moment in time where, are we really still Wednesday? Or is it Thursday? You know what I mean? So this could be the beginning of prophase. You could argue maybe I could start seeing some of the chromosomes, But definitely the nuclear membrane is still there. So I, could probably argue that it's most likely this is interphase. Okay, this looks like the cells are splitting in the middle. You can see there's cell, it looks like it's pinching in here. So I would say this is cytokinesis, telophase and cytokinesis. This is, for example, in the onion root. So you could see most likely this is interphase. Here I can definitely see the chromosomes. So this has to be prophase now because I can see the chromosomes, and the nuclear membrane at this point will start to break down. You can see metaphase beautifully here in this slide in the middle. You could see the beginnings of anaphase, right? And you can say that maybe this is like late anaphase, but possibly early, telophase, like the example I said. When you switch from Wednesday to Thursday, there's going to be a point where it's going to be a little bit hard to tell sometimes if it's Wednesday or Thursday. And then you could see that cell plate forming. So this would be telophase, where you get the cell plate. You can see forming in between the cells. Remember, a plant cell can't pinch in the middle. So those are the stages of mitosis. Okay, so we reviewed interphase is the preparation, and then you have your 4 stages of cell division: prophase, metaphase, anaphase, and telophase. Now, any questions about those stages? No questions? Okay. So it's very visual. Sometimes it will be a little bit tricky to identify stages. Sometimes stages are very easy to see. Maybe sometimes, like I can clearly see that this is anaphase because the chromosomes have split. I can clearly see that that is metaphase. This one you could argue maybe this could be, it says labeled late anaphase, but if you said this is the beginning of telophase, how can I say that you're incorrect? So that's mitosis. I want to quickly just compare this to meiosis and show you what the difference is between the two. Now, I'm going to keep this as simple as possible. So remember, in mitosis, what you're trying to do is you're trying to make 2 cells that are exactly the same, right? So you have a cell and it has DNA in it, right? And then you end up with 2 cells that are exactly the same. Okay, exactly the same, right? That's what you're trying to do. You're trying to make an exact copy through mitosis, right? Now, meiosis it's a little bit different because you're not actually trying to make an exact copy. What you're trying to do is you're trying to make sex cells. So in other words, you're trying to make a sperm cell and an egg cell. So it's going to be a little bit different. There's a lot of similarity, but I want to just focus on some of the differences. So one of the differences is if I look at what happens in meiosis is there's a lot of similarity, like I said. For instance, the DNA gets copied, just like it does in mitosis, right? But the big difference is this. At the end of the process, we're going to end up with cells that are going to have half the DNA. Okay? Now, I'm going to go back to, for instance, this slide here. You'll notice that here you have, we said, no, I think the last time I labeled the dad the red one. So let's just stick with red for dad, okay? Let's just stick with that idea. If you remember, the DNA is a strand, and then we make a copy, right? That's what we did in mitosis. The same thing happens in meiosis, exactly the same thing. So if this is your mother set in blue, then it's going to make a copy. And that's what you're seeing here, right? So that I said that these two are identical. There's no difference between them, okay? These two are identical. Now, what happens though in mitosis is the cell will split. So at the end of it, what you should get two cells that have one red strand and one blue strand each. Okay, that would be mitosis. And in mitosis, you get two cells that are exactly identical. But that's not what you want in meiosis. You don't want to make identical cells. You're trying to make sex cells because we're trying to make babies that are different from other babies. That's the point. We don't want to make identical babies. We want to make different babies. So you'll notice that at the end of this, you end up with cells that are not identical. That's one thing, right? You'll see, for example, that if our original cell had two strands, the daughter cells in meiosis only have one. So that's one huge difference in meiosis. One huge difference in meiosis is you have cells with half the DNA. Okay? That's a huge difference. The other big difference is that there's another process that actually happens in meiosis that does not happen in mitosis. And it's a process called crossing over, all right? This is when you go to another dimension. And that's what you're seeing here. It's called crossing over. Now, what happens in crossing over is that the DNA that mom donated and the DNA that dad donated actually exchange little sections. So if we go back to the example that we did, we used a color to represent mother and a color to represent the dad, right? We said, for instance, red is dad and blue is mom, okay? Which, if I think about it, I should have went with the other way around, right? But anyways, if I look at what happened in crossing over, you'll notice something interesting. You'll notice that I have chromosomes that are still, actually, I don't see any of the examples in here. Yeah, they're all been rearranged. So you'll notice that the chromosomes that are on this slide are not all red and they're not all blue. They're actually part red and part blue. Okay, so you can kind of see that here. So imagine that we have this. So here are your two identical chromosomes, right? Remember, you make a copy. And these are two identical chromosomes because just like in mitosis, you have to copy everything. Okay, now the crossing over will actually, you can see it's actually happening right here, will actually give you a new combination. So if I have this blue chromosome, which I said blue was the mother, and I have this red chromosome, which I said is the father, and they exchange, do a little swap here, then what you end up with is, now you'll notice a red chromosome with a little bit of blue in it, and a blue chromosome with a little bit of red in it, okay? And then when they divide, you can see now I have more than, like, I have more possibilities. I can now get a completely intact red chromosome. You can see it's all red. I can see one that's red with a little bit of that blue. I can see one that's blue with a little bit of that red, and I can see one that's intact blue. Why? What's going on? Well, the idea is this. The idea of crossing over is to produce as many combinations as possible. That's the whole point of crossing over. It's to produce different combinations. Because this is all in relation to sexual reproduction. And if you remember in grade 9, you probably talked about asexual reproduction versus sexual reproduction. I'm sure that probably came up maybe in elementary school. And the huge difference between these two is in asexual reproduction, you make clones. I guess you can say mitosis is like that, because in mitosis, you're making clones, right? The cells are identical. But in sexual reproduction, you don't make clones. You produce unique, different offspring, right? So, you can, unless you're identical twins, your brother or sister from the same set of parents, not the same, because you're producing a unique set of DNA in that organism. So meiosis, the job of meiosis is to mix up the DNA through a process called crossing over. And that gives you new possibilities. Now, so you can see there's a huge difference now between meiosis and mitosis. One is the cells end up getting half the DNA, and the DNA is all mixed up because of crossing over. Now, why are we doing this? Well, because if meiosis makes sex cells, then the math makes sense. So let me show you what I mean. So we said mitosis is you take a cell, it has 46 chromosomes, you copy it, right? Now that cell will have 92, and then it splits into two identical cells of 46 and 46, okay? And that's great when you want to reproduce cells in your body, right? You want to fix something, you want to grow, that's great. That's mitosis. But in meiosis, we're trying to make sex cells. So it's going to be a little bit different. What we want to do in that case is we want to take a cell that has 46 chromosomes, okay? We're going to copy all the DNA, and make it into 92. And then what we're going to do is we're going to divide it actually 2 times. So we're going to divide it. Actually, let me draw it like this so it's a little bit easier to look at. Oops. Let me try eraser. Oh, no, I have to clear the canvas. Sorry about that. Let me try that again. Okay. So start with a cell of 46, copy that into 92. Okay. Then what we're going to do is we're going to divide the cell like this. So now we're going to divide it. So that looks like mitosis. But remember, crossing over happens too. So these cells are not actually the same. They're different. And then we're going to divide them again. So mathematically, if we split them in half without copying, so we're not going to copy the DNA again. So not copy DNA. Sorry, my English is no good. Then if we just split the DNA again, then you end up with 23 chromosomes in each cell. Now, if this is a sperm cell over here, and on the other side, we do the same thing. Copy the DNA, split them in half. Okay, and remember, crossing over is mixing things up. So we have new combinations. And then we split the cells again. And we get, in this case, we get egg cells. So these are egg cells. We can see now why we have to split it again. Because Now, if I take one of these sperm cells, which is unique, right? The sperm cell is unique because of the crossing over. So this sperm cell is different than this one, which is different than this one, which is different than this one. And this egg cell is different than this one, which is different than this one, which is different than this one, which is different than this one. Now, if I take any random sperm cell, let's say this one, and any random egg cell, which is that one, and I put them together, what do I get? Well, I get a zygote with 46 chromosomes in it, which is the same as what a human should have. That's why we have meiosis, is so that we can make the sex cells. I'm tired. All right, now, so just by the way, just so you appreciate the uniqueness of this all, the differences, I'm going to kind of zoom in here. So mathematically speaking, this is without crossing over, okay? So without crossing over. There are 23 pairs of chromosomes. So that works out to about per sex cell, about 8 million possibilities. So there could be 8 million possible sperm cells and 8 million possible egg cells. You put them together, that means mathematically 64 trillion possible zygotes. So here's a word of advice. If anyone says to you, baby, you're 1 in a million, tell them off, because 1 in a million, you're actually 1 in 64 trillion. Actually, it's even rarer than that, because this does not even include crossing over. Okay, so I was going to explain your assignment on cell division, but I think I'll do that next time, because that was a lot and that took a long time. So I think in the meanwhile, if you girls want, there are course outline questions you could look at in regards to cell division. I think I have some. So I'll do this next time, okay? We'll talk about there's a lab I put together on cell division, but it's going to take me a while to go over, and it's already been an hour. So I'll go over this on Monday. There's really no rush. So then if you're bored and you got nothing to do, what you could look at is you could look at your course outline. And your course outline has questions. Oh, by the way, should go back to, there is an online version of your book. I should have mentioned that. If you don't have a book at home, but I wrote the questions so you actually don't need a book. This is the link, and then these are the, I should actually stop recording. Let me stop recording.